When I first started talking about microarray, it was kind of quite a long time ago, maybe 2 years ago, and it was a question of everybody wants one. What are they? What do they do? What's the technology behind it? And I think it's, although it's not quite so much the case, the excitement that's been generated around this microarray platform that we have here, and that with George Robertson's enormous contribution, we've managed to get into the IWK and set up and functioning. I still think I'm legitimate in saying that everybody still wants to use one if they don't want to actually have it. So, Mike, oh, Lord.
That's not our color.
That is not,
It's fine.
that is not my color in any way, shape, or form. So, what is microarray? It's a solid support medium, high resolution, used to detect DNA sequences. It can also be used to detect. It also can be used to detect protein sequences, but I'm really not going to talk much about. I'm not going to talk at all about that. Not specifically protein bound on the array. So it is a It's something very akin to a microscope slide. I was kind of hoping I'd have one to show you, because it's kind of like a show and tell situation here. Not much bigger than a microscope slide. This little square here is about 1 centimeter square. That's where all the spots are. And many probes are attached there. Any number from a small number to a large number, 3,000 to 2.5 million or more. And longer or shorter probes can be used, and that will contribute substantially to the stringency with which you can ask questions, you can interrogate. For the purposes of this discussion, I'm going to use the terms chip and array pretty much interchangeably. They are used almost interchangeably in the literature. They can be called the DNA chip, genome chip, expression array. It's still based on a chip. DNA array. The only thing that's not quite generic is gene chip is an Affymetrix registered name. And the chip is simply the substrate. It is the basis. The various chips differ in how they're designed to answer specific questions. What molecules are put on them, how many of them, what kind of stringency you want in the questions that you're asking. So the probes on the chips represent sequences of known sequences of interest. They can be large or small. Initially, chips used BACs, which are traditionally large molecules, greater than 100 kilobases. And they look at large regions of the genome. And I've put large in quotation marks because if you're a cytogeneticist, a large 100 kilobases is not a large region of the genome. It's a tiny, tiny region of the genome. But I'm a molecular person, so I think 100 kilobases is pretty big. And the BAC arrays usually have, because of this large chunk of DNA, they usually had lower resolution, and that was related to the size of the DNA that was actually on the array. So from BACs, there's a variety of intermediate steps, but now people are using all of those much more often, short pieces of DNA, maybe 25 to 70 bases. And they look at teensy, I had teensy-weensy, but I was told I should take it out. Teensy regions of the genome really as small, less than 1 kilobase easily. So not sequencing, but we use SNPs, and SNPs are by definition a single nucleotide. So they are generally associated with higher resolution, but it does depend on the array construction. If you've only got 1,000 spots of regions of interest, then your resolution is those spots. There's lots of DNA, genomic DNA that you're not looking at. When you have 2.6 million, which our Cytoscan HD array has, and which is close to what the SNP chip had, you've got quite a high resolution. So as I've already alluded to, we can easily see 25 kilobases, a resolution of 25 kilobases, and whoops, we can go, how did I do that? We can go down to less than a kilobase quite easily, but you get fewer probes within that small region, and the fewer probes you have, your confidence in that call becomes less and less. So you can go smaller, but you lose the specificity, or you have decreasing accuracy of call. You can use microarrays for, I can't see in this slide, okay. You can use microarrays for a variety of, to answer a variety of questions, or to ask a variety of questions. You can look at expression arrays for RNA analysis, and that I've just kind of loosely put them into two categories of kind of an absolute question, that is to say, what genes are expressed when and where, at what developmental stage, what tissue, what organism, or you can do relative, where you are comparing a control, two different samples. It could be a abnormal to a normal, or it could be any developmental stage to a differentiated cell, or stages in tumor progression, or pretty much anything that you want to ask. So that's all RNA-based. Family studies, looking at linkage or haplotype, such as identity by descent and homozygosity mapping. These are traditional uses of the SNP chip protocols. And now we know we can, with SNPs, we can do dosage analysis, so we can look at copy number variations. And this is very important in clinical analysis, and Karen's going to be presenting 3 clinical cases to illustrate how we use copy number variation. And the dosage is done by SNPs and the non-variant oligos that are also on the Cytoscan HD. It's not all SNP, it's about half. I'll break it down a little bit later, but it's SNP and non-variant oligos. So 2 categories of probes that are on the array that are non-equivalent. Resequencing, microarray was probably the first commercially available platform to be able to sequence in parallel a variety of different sequences of interest, genes, panels, whatever. At the time, it has its place. It is still used, but for general resequencing purposes, microarrays are really not used at this point. They're too limited now that next-gen sequencing approaches are so well developed and commonly available. But certainly, microarrays such as the one we're using could be used for resequencing, for mutation identification, either known mutations, a known panel of mutations, or to actually sequence generally, say, affected individuals with unknown genes, as in exome sequencing, or targeted sequencing, known affected individuals, known genes. What? Well, you don't like the color.
There you go. A little bit better.
Ah, good. Don't touch.
It's the cable.
Okay, I'll try not to touch. I'll leave it hanging off. Okay, so back to the array. It's a specially treated glass, nylon, or similar material, very similar to what they use in making computer chips. The DNA representing the region of interest is bound to specific positions, and AFI uses photolithography to do it. I've got a slide that just shows that a little bit. At each point where you put, at each spot where you put sequence, there are multiple copies of that sequence at that spot. And sequences at different spots may differ by 1 base or 100%. Depends on what you want to put there. Depends on the stringency of the question that you're asking. For the Cytoscan HD chip, which we use, all the sequences of interest are represented on one array, usually at multiple spots, so that there is redundancy built into the system. And as I say, you can pack a lot of information into 2.6 million slots. So I'm not going to spend a lot of time here. It's a sequential photolithography, so you bind a reactive molecule to the chip, you put a blocking compound on it, you put a mask on it, you then activate what's not masked. And that releases the blocking agents. You then add the base that you want. You reblock, you remask, you reactivate, and you keep going until you have built up at your various spots the sequences that you want. So very much like building up a computer chip. These are pristine facilities that do this. And the wafer, this is a wafer, and although it looks like a pretty big piece here, each of these little squares on the wafer is 1 centimeter by 1 centimeter, and you get 49 off one wafer. So when you want to buy a kit, you buy 49 of them. Well, you actually buy 48 of them because one's used in quality control. So just an FYI. So the sample, I'm going to go a little faster here. I've already taken more time than I was supposed to. Sorry, Karen. The samples of interest are treated to make them amenable to hybridization. So for expression arrays or expression questions, you have to convert the RNA to cDNA, then make a sense cDNA, then you label and hybridize. The rest of the molecules, like in DNA analysis, you are going to be fragmenting, labeling, and hybridizing. The array comparative genomic hybridization, you have two different samples coming through. One is labeled with one dye and one's labeled with another dye. You competitively hybridize them and then put them on the array. And that's the only difference on that one. And resequencing, same thing, fragment, label. Sounds very simple. It's not really quite that simple. And the reason I put down hybridization overnight here is because this is where the microarray platform starts. So everything else is workflow that is common in any molecular lab. So you hybridize, as in most hybridizations, you remove the excess by washing, and then you scan, in this case with the laser. So this is the rest of the microarray station, the fluidic station, which is basically a robotics, and the scanner. And in the scanner, the chip is held in place so that the scanner can read each spot and know exactly what spot it's reading, and it knows what the sequence is on that spot, so it's reading the fluorescent dye that's at that spot. So it determines what sequence each spot represents, assembles the results into an ordered or sequential data set, or overlapping regions which may or may not be contiguous depending on the question that you're asking. And dosage now, because of the technology is so sophisticated and so well controlled, we can take the intensity of fluorescence and say that represents the dosage, the copy number of the starting molecules. So originally, when we were doing CNV analysis, we used the back arrays. They're fairly limited. They detected a small copy number variation, 1, 2, or 3 copy numbers. And they couldn't look at balanced rearrangements. They historically were not able to pick up UPD. They didn't have any polymorphic content that they recognized. And they were designed, the arrays were designed so that there was a backbone, fairly minimal, of the genomic coverage, like every 10 kilobases, every 25 kilobases, and then a little more coverage of clinically relevant regions. And on the research side, there was the SNP chips, which have been used for years in gene discovery projects, high density, single nucleotide polymorphism chips, and well used in the research community. And as technology of the SNP chip analysis became improved, it was apparent that you could start reading dosage off these. And this was a real realization, because it was not felt at first that it could be quantitative. But once it was realized that it was quantitative, then people who thought dosage could be important, like low copy number dosage, like 1, 2, 3, 4 copies, they got very excited about it, and they started using SNP chips, exactly the same SNP chips, for copy number variation in cytogenetic analysis. So lots of advantages there. You can use their oligos, so you can put any probe on it. Very fine resolution. You could pick up heterozygosity. This was pretty exciting, because this was a whole area of information that wasn't available earlier. And because you could pick up heterozygosity, you could also pick up absence of heterozygosity, which is important in human genetics, but it's also important in understanding the molecular pathogenesis in oncological transformation. So it's not tailored. The disadvantage was the SNP chips were not tailored for cytogenetic regions of interest. It was designed so that the SNPs came from across the genome. And a new generation of SNP chips now dedicated to copy number analysis, this is one that we use, Cytoscan HD, is now industry standard. And many other companies are starting to generate these SNP-based chips. And I say SNP-based because you still have non-variant oligos on there. And they still cannot detect balanced rearrangements, which for cytogenetics is pretty important. But because of our very fine resolution now, the question is, are these rearrangements that are described as cytogenetically balanced, or described cytogenetically as balanced, are they truly balanced when you start looking at the molecular ends of the breakpoints? And there's some indication that quite a lot of them really aren't, that you can actually pick them up. So what this doesn't do is it doesn't tell you where your copy number variations are. This tells you that they're there in the genome. So that's, for clinical application, that's very important. And for oncology interpretation, that's important too. So if you have duplications, we tend to say, oh, the duplication is right there in the same spot, but we don't know that. It could be somewhere else. So this is the chip that we use. About 2.6 million, of which approximately half are SNPs and the others are non-variant oligo backbone. And this is their distribution. So in the cytogenetically relevant area, one marker every 384 bases. For the genes, that's online Mendelian inheritance in man, for people who aren't geneticists out there. The coverage is about one marker in every 880 bases. The same for the ref sequence genes. And the genomic backbone is much slower density, one marker every 1.7 kilobases. So very much focused on regions of clinical relevance for the chip that we use. There are many chips available. So I'm going to hand my talking stick over to Karen now. I'm not sure, I don't have time, so I'm not sure whether we have time for this little video. Somebody tell me what time it is? 12:34. Oh, I think we'll skip that. We'll just go straight to Karen. You have to kind of judge that here, Karen. It's your turn.
Thank you. Sorry, I just have to load this on. I hate using the mouse. Okay, so for the next 20 minutes or so, I'll very briefly talk about the clinical applications of microarray analysis. We have gone live with microarray testing at the IWK this past week. We have spent probably the last 6, 7, 8 months preparing, doing an extensive validation. We've done a lot of training, worked with a lot of cases to get the experience with trying to be able to interpret the test results that the system gives us. So what I'm going to do is talk about the clinical indication for microarray testing. It is not a population screening test by any means. It's a very expensive test, and so there are certain clinical indicators where it's recommended as a frontline first-tier test. I'll talk about the clinical requirements of the array platform. As Christie so nicely outlined, this particular array platform that we're using can do a lot. So there are certain criteria that need to be met in order for it to perform in a clinical setting, and they're usually quality indicators. And I'll talk a little bit about the categorization of the microarray test result. What is a CNV? What are the different categories of CNVs? And what information does the SNP tell us when we're applying it to a clinical test? And I have 3 case examples, because I think it's always interesting to see the actual, they were teaching cases, part of our validation that we were doing at the IWK, and then just to see how we actually use the data. So traditionally, when we have had indications for intellectual disability, multiple congenital anomalies, dysmorphism, usually our frontline tests were doing a karyotype to see if the karyotype was normal or if there was an aneuploidy there or a structural rearrangement. And sometimes we would also shunt over to targeted FISH if we were concerned that there was a microdeletion present, something that we couldn't see at the cytogenetic level. The advantages and disadvantages of these 2 technologies is cytogenetics will give you a genome-wide screen. You can look at every chromosome. You are down to a single cell resolution, but our molecular resolution is about 5 to 10 megabases, which is roughly 1 chromosome band. So if you can see a band that's present or it's not present, fine, but if it's something smaller, and a lot of the pathogenic imbalances, they are much smaller. The other thing with FISH is it gives you a much higher sensitivity. You can find those submicroscopic imbalances, but it's targeted. You need to know, you need to ask the question, what probe, what is it that I'm looking for? What region of the genome am I going to investigate? So it's not a whole genome-wide screen. So those were the limitations. Together they're very strong, but certainly we knew there were a lot of smaller imbalances in the genome that are associated with disease. So in terms of the clinical indications for microarray testing as a first-tier frontline test, there are basically the 4 indications of developmental delay, intellectual disability, multiple congenital anomalies, and autism. And the microarray becomes cost-effective if this is done in lieu of karyotyping and some of the other targeted molecular tests like Fragile X testing and so forth. That's where it becomes very cost-effective because it has a much higher resolution and a much higher pickup rate. What it's not recommended for is the common aneuploidies. So if an individual is suspected to have trisomy 13, 18, 21, there's mechanistic information that we need to know about that aneuploidy. So either doing a very rapid molecular aneuploidy screen as our first test to see if there's aneuploidy there. It's cheap, it's quick, it can be done in a clinical laboratory setting. And then using conventional cytogenetics to see, is this actually due to a straightforward trisomy, or is it due to a translocation, an unbalanced Robertsonian translocation, which has implications for recurrence risk for future pregnancy. It's also not recommended for the sex chromosome abnormalities. The array that we're using, because of the SNPs being added into that, I think it has a little more power to look at sex chromosome aneuploidy, but a lot of the sex chromosomes, they can also be, we're looking at balanced rearrangements with autosomes and so forth. With the balanced chromosome rearrangements, when we get a clinical indication of infertility or recurrent pregnancy loss, microarray is not the test of choice for that. You would still want to do conventional cytogenetic testing for that, because you want to be able to see those translocations, and that's something that microarray will not always pick up. The percentage that Christie had mentioned, about 45%, are actually unbalanced at the chromosomal breakpoints of a translocation. That's in a population of developmental delay, and often it's a de novo chromosome rearrangement that has not been inherited from a parent. So there's some pathological underlying mechanism that's associated with what appears to be a balanced cytogenetic translocation. So in terms of the microarray platform requirements, there are guidelines that have been published by the Canadian College of Medical Genetics and the American College of Medical Genetics that says for clinical use, a microarray platform should be able to detect a minimum imbalance of 400 kb. So that would be a loss or a gain. And the way that that's achieved is through a very high density probe coverage, as Christie outlined in the slide, with something like the Affymetrix system, and a whole genome coverage. So you want to have the whole genome, excellent coverage. You also want to have very dense coverage of those regions that we know are clinically important. They're dosage-sensitive regions that we know are known to be associated with a particular syndrome. And so that will give you the minimum 400 kb resolution that you need with a sensitivity and specificity of 99% or greater. And it also allows you to pick up those very tiny CNVs that are within the clinically significant regions, being able to combine the two. In terms of the regions of clinical significance, what most labs are currently using now is a combination of the international standards for cytogenomic arrays. We call it ISCA. It's going to be changed to ICCG, the International Consortium of Cytogenomics. I forgot now the new. It's going to be coming out in the new year under a new name. What we have, the ISCA gene list is a list of genes that have been defined by the ISCA consortium as being clinically relevant. So these are genes that we want to look at practically at a 0 kb level. So what is right down to the lowest or the highest resolution that we could go. And the other thing that we're using is the deciphered known microdeletion duplication syndrome list as well. So these are tools that we're using that we brought into the software to help us identify those regions of interest when we're doing clinical cases. In terms of the CNV categories, they have been around for a long time, and there has been a lot of attempt to standardize their definitions so that when someone says this is a pathogenic CNV, we are all working with the same definition. We don't have a different definition per lab. And the CNV categories that we're going to be using are actually published by the American College of Medical Genetics, and there are 3 broad categories. So they are pathogenic, they're benign, or they're uncertain. And the uncertain are broken down into 3 more categories that are likely benign, they could likely be pathogenic, or we just don't know. And often these CNVs with the uncertain clinical significance, they are 400 kb or greater. That's our promise to report. We're promising to report CNVs of 400 kb or more. And so we're using this classification for our CNVs. The other thing that the microarray tells us, as Christie said, is it gives us information about homozygosity, and you'll see this in the literature in a number of different ways. The AFI system actually calls it LOH, loss of heterozygosity, which is more of an oncology term. For constitutional evaluation, we're looking at absence of heterozygosity or something called long contiguous stretch of homozygosity. I just find AOH easier to say, so I'm going to stick with that. And the other nice thing, I do have a case with that, where it just very nicely shows these regions of homozygosity and the clinical relevance. The other very nice thing about having a SNP CNV microarray is if you identify a CNV, you can use your SNP information to confirm that that actually is a gain or a loss. So it's almost a second confirmatory mechanism. And the guidelines are stating that you may not always need another molecular or FISH or some other type of confirmation if your SNP clearly shows that your CNV has the same information. So in terms of the microarray, I think it's always important to remember what it can't do. So even though we have this wonderful coverage, we've got great probe distribution across the backbone, we've got very good distribution within the clinically relevant region, we have to remember that no microarray, it doesn't matter how souped up the platform is, will detect all mutations that are associated within a given region, even if it's thought to be well represented as a clinically defined region on that array. It won't detect your point mutations. It may not always detect your tiny CNVs. Balanced chromosome rearrangements, it may not detect all of them either unless there are submicroscopic imbalances at the breakpoint that are pathogenic. The other thing is there are still gaps in the genome. So there are regions where there isn't the greatest probe coverage, and largely that's due to your repetitive elements. There are your segmental dupes, it's your genome architecture. It just makes it very difficult to get reliable probes for those regions to give you reliable readings because you don't want spurious results. So what I'm going to do is talk about 3 case examples, highlighting how the system can be used to identify a small microdeletion, mosaicism, and also AOH. So the first case was referred for developmental delay and dysmorphism. And when we look at the microarray software, it's a very, very busy screen, and a long part of our training working on this together, which is getting used to all the information that the software gives you. And I'll be, Christie and I will also be giving a couple of seminars with the clinical genetics group. So I'm going to go through this quickly because we don't have a lot of time, but we'll go through in a little more detail with the clinical group when we meet in January. So basically what the software does, it allows you to set up all of your filters. It gives you a karyotype view, an ideogram view of your microarray results. It gives you segment tables, it gives you cytoregions, it allows you to find an overlap map. So this would be a map where we're saying, these are regions of importance, pay attention, highlight them for us, flag them for us. So, and then we have additional individual probe information. So when we're opening up the software and looking at the CNV, first of all, what you can do, any CNVs that are listed, and a lot of these are tiny, so we have little small red triangles that are losses, and small blue triangles that are gain. And what I like to do is I usually go to the chromosome view and I look and see are there any really large imbalances, because sometimes it can be an unbalanced translocation. And you'll see that very, very quickly with this view. We have ideograms over here. Most of the information that's on the right of the chromosome is pertaining to that sample. All the bars and the lines that you're seeing on the left of the chromosomes, they're the different maps or the different regions that we have defined as saying they're important. We want to see them and see how they connect with the CNV that we're looking at. So this, when we do a further evaluation, this is what we call our detailed view. So we have the karyotype view, and then we have what's called a segment table. And what I've actually done is I've tried to maximize the detailed view and take you through the CNV review. What the segment table does, it will list every CNV that the software is called. It'll tell you, is it a loss or a gain? Is it, what is the copy number state? What chromosome is it on? What's the chromosome band from start to finish? The number of markers that are involved in that CNV, and the marker number is actually important. So there are minimum criteria of number of markers involved for a CNV call. Your max min, You've got your chromosome position of where it is in the genome, your size in KB, and then we have additional information here. This just stretches on and on, where we have multiple columns showing the genes, the OMIM disease genes, and many other QC checks that we go through. So this is a particular CNV that I pulled up because I wanted to show you how we go through the process. The red bar here shows you the CNV. We have log-to ratio information. So this is essentially the distribution of the probes along a graphical image here. And most of the time when there's normal copy number, these probes or these points are just going to go along this middle line here. But if we start to have gains or losses, they're going to deviate from that middle line. So here we have clearly probes that are below the line. We have very good CNV probe coverage. So there are probes that are distributed all across the CNV, and that becomes important because if there are regions of segmental dupes or repetitive element, you may not have as many probes covering that CNV, and you might get a spurious call. The software will call it, but we still do a visual evaluation of that log-to ratio and that CNV. We also look at what's called a smooth signal. This allele peak track, when it's a really tiny CNV, is not as informative, but I have some other case examples where you see the allele peak track really becoming a useful indicator. But in this case here, our smooth signal line has dropped down from 2, so it's gone below. And so we've got a very, very nice CNV call here of a small 70 kb deletion. So it's real, we're happy with the data, we're happy with what it looks like. The other, we have a lot of information on the screen here that becomes important for us when we're doing the CNV interpretation. We've added some extra files to the system. It allows you to import what's called AED file. And we have our ISCA constitutional regions. They're all marked. We usually, with a brown rectangle, we've got our decipher syndrome list with the green bar. We have our Toronto database of genomic variation CNVs. So these would be CNVs that would be seen in a normal population. We have our genes that are involved here. We have our OMIM, morbid disease genes, showing up in the bright green. We have placement of segmental dupes, and we have our marker distribution. And down here we have our chromosome band, which we can, as a cytogeneticist, I'm always looking at the side here to see where does that relate, am I in the right position. So right away, I know this doesn't come up very clearly, and I apologize for the resolution. The only way that I could get this is through a screenshot, a print screen. But this little ISCA bar is telling me that this is the Wolff-Hirschhorn microdeletion syndrome critical region. So again, it's a region of interest. Clinically, it's quite relevant. The decipher tells us that this green bar here is the Wolff-Hirschhorn syndrome deletion region. So this CNV is falling within these two clinically relevant tracks that we have, and it's also here overlapping with our own disease gene map. So right away we've got a lot of evidence for pathogenicity for that CNV based on its location, based on its overlap with known microdeletion syndrome region. So again, that we can get some information very quickly. The other thing that we do, there is an icon up here in the toolbar here that will allow us to link out directly from the software to the UCSC browser. And what that allows us to do very, very quickly through the UCSC genome browser is we've set up our page so that, again, we get a lot of clinically relevant information. We have our gene content, our RefSeq genes. We have our OMIM morbid map disease genes coming up. We have our ISCA CNVs. So these are CNVs that the ISCA consortium has defined as being either pathogenic, benign, unknown. Some of them are curated. They're trying to curate as many of their CNVs as possible. There is also the Toronto database of genomic variants, segmental dupes. We can link out to the decipher database very, very quickly. We can link out to NCBI and get our gene list. We can find out what the genes are, what they do, are they involved in any kind of disease, and we can also link out to gene reviews. So that's all very quickly done from just clicking an icon in the software and linking out into UCSC. So for the case 1 summary then, what we've identified is a very small CNV, 70 kb, that contains the WHSC1 gene that falls within the Wolff-Hirschhorn critical region. Wolff-Hirschhorn syndrome is a contiguous gene deletion syndrome. It's roughly about, if you look at the chromosome position, we've identified a very tiny deletion within that region. And it's roughly, on average, about a 2 megabase deletion, but the size will vary. The clinical features are quite distinctive. You will have the severe growth restriction, the intellectual disability, your microcephaly. You have a very distinctive facial presentation of a slanting forehead that they call the Greek helmet face. And there can be closure defects as well. So when we actually did phenotype-genotype correlations, this patient did present with Wolff-Hirschhorn syndrome, and we were able to identify this very, very small 70 kb deletion that fell within the Wolff-Hirschhorn gene. So as part of our validation, it was very important that we be able to pick up those tiny CMVs that are associated with disease. We needed to know how the system worked, and it gave us a lot of confidence that we were going to pick up the very small calls, and that we had our parameters and our filters set up appropriately. So the second case, again referred to for Deaf delay dysmorphism, autism spectrum disorder. The array identified, and this would be from the segment table information, 195 kb gain on the distal part of 19q. But when we looked at the smooth signal, it wasn't a complete gain. If we look here, this is our entire CNV shown here in the blue. So I'm looking at probe distribution across. It's not quite all above that line, like it was in the previous where we're going up or down below our middle line, 0. And if you look at our smooth signal, it's hovering between 2 and 3. And that would suggest that we might have mosaicism. That is not a complete gain. If it would have been a complete gain, we'd be looking at 3 copies. But we've got between 2 and 3. The log-2 ratio and the smooth signal both kind of support that. So what we were able to do then is, when we're suspecting mosaicism and we've got a small 195 kb call, is we can just zoom out. So we can look in detail at the CNV, but we can also do a whole chromosome view. So this is the entire chromosome 19, the log-2 ratio, the allele peak tracks. But what becomes really informative is that smooth signal line. Do you see how it starts to go up above 2? And it hovers between 2 and 3. And what we finally estimated this to be is a much larger gain, a mosaic gain that involves the distal end of 19q. So our segment table, we're taking the information from multiple places. So our CNV segment table call with 195 kb, our smooth signal suggested that we have mosaicism, and we can use all of that information and put it together and say this is what we think is going on, that what we've identified. So we have roughly a 5 megabase mosaic gain. Certainly with something like this, you would want to have follow-up confirmatory studies, doing chromosome. Some labs with the microarray time, just telling you time, they may do fish as well. So the last one, very quickly, and I'm almost done, is, but you're going to be short.
It's okay, keep going.
Do you want me to keep going?
Yep.
All right. So this was a case just demonstrating AOH. It was a carrier of a balanced Robertsonian translocation involving 13 and 14. And whenever you have chromosome 14 involved in a Robertsonian translocation, there is a concern for uniparental disomy. When we looked at the SNPs for this case, we saw quite clearly a very large region of AOH on chromosome 14 that was almost 50 megabases. So it was very clear in the karyotype view. If we went to the allele peak track, you can really see this quite nicely, where in a normal, when you have a heterozygous state, you have your AA, AB, and BB tracks, and here you've lost your middle track, showing you your homozygosity. Very, very easy to see, very, very quick. So the nice thing about the SNPs, as Christie kind of set the tone for, and we'll hopefully talk about a little bit in your Andy, we'll see. We can get a lot of information. We know what's normal, what's a loss, what's a gain, what's an AOH, and mosaic. So we use this as a crib sheet, actually, when we're looking at these allele peak tracks, to give us some more information about what we're seeing. So in terms of the case 3 summary, almost 50 megabase AOH, you have to think about UPD14 because it is an imprinted chromosome. You can have, depending on the parent of origin, maternal, paternal, different phenotypes associated with it. Whenever you have regions of AOH, you have to have a concern about uncovering an autosomal recessive disease. And also, in some instances, you may have residual or very low level trisomy 14. And you would have, again, for this kind of a case, you would have a follow-up. So in summary, we can identify CNVs, small deletions, small gain. We can, in some instances, identify mosaicism. We can identify regions of AOH. So it gives us a lot of information. And I'll stop right there. I think that's a good place to, because of time. Just while I'm trying to get out of here. And show this is what I want to do. Okay.
So, organizers, what happens with the time? Do we all disappear at 1 o'clock?
We usually try to close it at 1. If people want to leave, you can leave. But if you want to stay for questions and do the rest of what Christine has to say,
Okay.
Karen.
So I have about another 10 minutes if people want to hear that. It's on other applications. Or we can start questions now. I can talk really fast, or we can just answer questions. Talk fast. Okay. I'll talk fast. So, there are other applications of the platform that we use. Okay, there are other applications of this platform that we use. We can, there are chips dedicated to looking at tumor tissues. They look at copy number, they look at known mutations, or you can do RNA expression analysis. So there is an oncochip dedicated to hematological oncology samples. RNA expression, I'll talk about down here. So there's several different kinds of RNA expression analysis. Traditionally, it was the 3' IVT or the 3' UT arm. Most of the transcripts were looked at from the 3' end, and that was kind of early in the technology. Now you can look at the whole transcriptome, and there's 2 different versions. You can look at it with or without the line C mutation RNAs, and there are also arrays that look at microRNA. So the arrays that are being supplied by the company are increasing in number and in detail, and you can also custom design arrays. I'm going to spend a minute, literally, on this chip-on-chip because I think it's kind of interesting. So this is the closest I'm going to come to talk about how you use the chip to look at protein interactions. So you've seen a couple of pictures like this. Now I'm just going to go through them quickly. You now know all the critical, the key places on this screen. What I want to show you is this is the SNP allele peak that you can clearly see homozygous along here, homozygous upper, homozygous lower. This is the heterozygous peak. And here it changes. The log 2, the smooth signal, has this jump right here, and it doesn't actually, it goes from 2, which is normal, to not quite 3, so it's mosaic. So, mosaic gain, this is in a CLL sample on chromosome 5. All these little red bars, well, they're actually all red, even though they look kind of purplish there. Each one of those is a gene. So you can see there's no real difference, they just separate them out because of the number of genes that are involved, that are displayed. So there's lots of genes in this region where there is this mosaic gain. So this is in one chromosome, chromosome 11, mosaic loss, mosaic copy neutral, loss of heterozygosity. There's your normal floating along here. You can see the 2 outer flanking lines get pinched into here, and the inner heterozygous line splits into 2. And then they go back out to a line that's pretty equivalent to the normal homozygous, and down here the normal homozygous. So this is the mosaic loss, where you get a pinching of the outer bands, and back to copy neutral. So there's still 2 copies there, but it's now not a homogeneous population. There's 2 different populations, and so that's where the mosaicism comes from. And I said that's on chromosome 11. And another mosaic loss and mosaic gain on chromosome 8. And again, the striking change here, you go from the loss here, look at that signal. This is 2 right here. So not quite 1 and not quite 3. And you can see how the patterns change. This is chromosome 8. All 3 of these slides are from the same CLL line. So many changes involved in the various lines. So you have copy number information, you have heterozygosity information, and if you have a lot of heterozygosity, you may ask, what is this heterozygosity unmasking? And so this is an article from 2009 where a variety of genes were shown to be unmasked by acquired somatic, they call it acquired somatic UPD, I'll just call it loss of heterozygosity. And these genes, mutations in these genes were characterized to be homozygous in these particular examples. And so the suggestion is that maybe there's more to come. So there's a huge amount of, this is copy neutral LOH. So that means that it's like a UPD thing, the two copies are the same. 20 to 80%, well that's a big range, but here the numbers here are 20 to 81% in these cancers here. And this is an Affymetrix plug because all of these arrays are Affymetrix arrays. And I'm not going to go over it because of the time. There's a variety of arrays which allow you to ask different questions about the tumors that you're looking at. And if people want more information on this, we can come back to it. So I'd like to spend a little bit of time on expression arrays. They've been used for a long time, 12, 13 years now. And traditionally they looked at the 3' UTR and transcripts, the transcripted sequences that are close to the 3' UTR. Now we know that this is not representative. It was what we could use at the time. It gave us a lot of information, but we now know it's not representative. There's multiple transcripts per 3' end. Not all 3' ends have poly A tails, therefore they don't get amplified. And there's a variety of other drawbacks to them now. So the expression array technology has been dramatically improved by the use of oligos. So it's a very similar kind of chip, except that it's looking specifically at transcribed regions. So we have better coverage, sensitivity, and specificity. We have much better basic knowledge, molecular genetic knowledge, on which we can integrate the expression information that we derive from these chips. So everything has progressed enormously in the last decade. The goal, at least the Affymetrix goal, is a minimum of 1 probe per exon. And if you'll remember that some of these kits involve the line cRNAs, so we're not even talking about coding, only coding information. I'm not going to spend a lot of time on this. There's two ways of looking at RNA expression. One is to compare an experimental to a control, differential labeling of the RNA from the control cell and the experimental cell, and you do the competitive hybridization, put it on the array, and then you get this kind of heat map that comes out of it. And if your control is green and your experimental is labeled with red, so Cy3 and Cy5, excuse me, then when the genes in question have the same level of expression, you get yellow, you get the green and the red, you get yellow. If the control has a gene that is, if the experimental has a gene that is that is downregulated, then that will look green in this heat map. And if the experimental has a control that's upregulated, so there's more red, you'll start seeing red. So each of these spots is a known sequence. The computer puts it together and tells you what genes are upregulated or downregulated, and you can distinguish to a certain amount the extent of up or down regulation. So the gene chip expression array, which is what the Affymetrix chip would be, is RNA taken from a particular cell type, whatever the cell type of interest is. It's extracted, it's reverse transcribed, it's labeled, it's hybridized, it's fragmented, so it's an appropriate size to hybridize to the chip, and hybridized, washed, scanned, and you get a much more complex scan coming out of the end. And again, the software puts this together into whatever format you want to look at it. Is it a sequence? Is it a series of contiguous positive responses, or is it a heat map which you've seen where you correlate tumor type or developmental tissue type with the genes that are being upregulated or downregulated? So this is the chip that would change whether you're looking at the 3' IVT, the whole transcriptome, microRNAs. This is what would be the chip. The chip would be developed specifically to interrogate the cells with the question that you are interested in. So the chip on a chip, chromatin immunoprecipitation, and it's used to enrich and identify genomic DNA sequences which function by binding regulatory proteins. So the genome, the source tissue that you're interested in, you cross-link the DNA and protein and fragment it and purify it so that you now have this cross-linked DNA protein. And depending on what it is you're interested in, you ask the question, is it histone? Is it histone binding DNA or acetylation? Is it transcription factor binding sites? Are they scaffolded proteins? Whatever it is you want, where the DNA and the protein actually interact and get cross-linked. This provides you, because you can now look at those, this provides you with a way to map those regions, those DNA regions, in an expression-specific manner, because obviously we tend to think of, all right, you have a gene, you have a methylation, you have a promoter, you get methylation at the promoter end of it, therefore you'll get that gene. But in fact, that gene obviously changes depending on the tissue type that you're looking at, the developmental stage. And we now know that there's a huge amount of regulatory information in the intervening sequences in the line long interspersed non-coding RNAs between genes in the intergenic regions. So this provides a novel way to map gene regulatory sequences in an expression-specific manner. So this is so you immunoprecipitate with the antibodies of interest once you've cross-linked your DNA, and I've already mentioned these. And the arrays that you use are high resolution, 35 base pair resolution, and you ask the question, where do your DNAs that you have cross-linked to the proteins, where do these DNAs map on these arrays? And then you just correlate with whatever your question was, the genotype, expression, copy number variant, alternative splicing, or whatever. And just to show you that it does give you interesting information, so this was just a little pie chart that I pulled out, so I thought it was pretty clear. So this is transcription factor binding sites. And I'm sorry, I don't have the tissue type that it came from. But basically, when they mapped all of the regions that were cross-linked to the DNA from DNA that was co-precipitated with antibodies to the TFBS, to the transcription factors, they came with known genes, 5', 3' are flanking to a known gene, and then a huge chunk of extra information, novel sites and pseudogenes or ambiguous binding. So this is, I mean, even if you just look at this, that's a full 30% increase in number of sites that now people want to say, well, why? Why is that important in this cell type that I was looking at? So, that was less than 10 minutes, wasn't it? So, quick acknowledgements. I'm sorry, Karen didn't get to put her acknowledgements up, but I'm going to. So, top of the list is George Robertson because it's his platform. And it's a great arrangement that we have, a relationship that we've developed, and I hope it's a long and healthy one. Thanks to George for letting us share his toys. Thanks to Alan Horsberg, who's the VP Finance at the IWK, for seeing the wisdom of letting the microarray come into our genetics diagnostics workflow. Tim Mailman, who's head of the lab medicine at the IWK, for putting up with all the machinery. Well, George and I were wrangling the details of getting that microarray machine moved and installed and functional. Stacy Thompson, and Cheryl goes here, but when I did this, I was really just thinking of who developed the basic techniques. Stacy Thompson, technologist extraordinaire, who generates, with Cheryl's help now, incredibly beautiful results. And Sam from Affymetrix for guidance with the complex software and for providing at least a few of the slides that are in my talk and also in Karen's. So, and since Karen didn't get a chance to do it, I'm going to say one more thing. Thanks to Hutton Kearney, who's the gal who allowed us to learn at her side how to interpret the data that we get from the clinical CNV analysis that we've been doing. So, that's it.
Any questions?
George.
Yeah, I should just say a couple of words on behalf of our sponsors. So the equipment was put in with assistance from the Department of Psychiatry, and also we were able to get a grant from a cohort BP program. Ray Roberts is here, and I would be remiss if not thanking him and Shane for the efforts they went to getting it, as well as everyone else. So we're hoping to put a users group together now that Christie and Karen got the workflow sorted out, we'll be in a better position to do further training. So I guess we'll let people know when that's about to take place.
Anybody who has research projects, talk to George. He's the research side of this.
Yeah, so I'll try to organize something over the new year around the so stay tuned.
Thanks, George.
Andrew?
Christie, what's the rough cost of one of your experiments?
A full run, we haven't actually worked it out yet, but it works. One chip analysis probably is in the vicinity of $500 to run. But when you do a full run, it probably drops to about maybe $350 per chip on a full run. So batch discount, but that would be, we haven't decided. Leanne's probably crying wherever she is over there.
Yeah,
But because that doesn't include overhead or things like that. So that's really off the top of my head. The chips are $200 a piece. Yes.
How long does it take to run through the experiment to get results?
The RNA expression array workflow is about 3.5 days, and the DNA workflow is about 4.5 days. And you could stop. You can carry the workflow forward until you get to the until you're ready for the hybridization. Once you start the hybridization, you have to keep going. Up to that point, there's a variety of places you can stop in the workflow. And I don't know about the RNA expression, but we have an autoloader on the scanner. We have one fluidic station, so only 4 chips can go through at a time in the fluidic station. And I suspect that this would be restricted even for the RNA, a bottlenecking for the RNA analysis. The autoloader will allow you to load 48. We can put, so if we can get 48 chips to the scanner stage, we can load 48 in the autoloader. And it takes about a half an hour a chip for the DNA, and I'm going to say probably about the same amount for an RNA chip analysis.
There's one question there, and then Jason.
Just a critical question. Because it's a SNP array, should we be cautious if we're ordering this on patients who are the product of consanguinity? Is that going to delay results?
Is that important for the clinical question you're asking?
I mean, it could be in some cases.
I think you have to know that we can look at homozygosity. And you will know once we go through the protocol in early January. So the question is, if that homozygosity might be relevant for the clinical issue that you're looking at, then yes, you have to get it.
Because sometimes we have these kids, like, we might be suspecting a recessive condition, in which case that could be helpful. But at other times, they're just a kid with developmental delay, and they happen to have
homozygosity might be part of that developmental delay. If the consanguinity is such that it concerns you, then probably you want to know the answer. And we're only going to report regions that we think are relevant, sizes that we think are relevant, sizes of homozygosity. So list your questions so you can ask us in January.
Hi, Christie. Thank you and Karen for doing this. It's a great overview. I had 2 questions. Just briefly, one is, in terms of sort of using other chips, like we want to look at zebrafish, for instance, does this platform allow you, if we purchase a chip or working together with you to purchase a chip, to sort of run that?
It has to be an Affymetrix chip.
Yeah.
You can have Affymetrix chips tailor-made.
But it has to be
But you won't be buying them by 1 or 2. You'll be buying them by
in a batch.
In a batch, yeah. But yes.
You can do that. And then the second question is in terms of you talked about a lot of technologies here associated. Certainly from a research point of view, there's a growing interest in doing RNA-seq as well. And I don't know if you could just for a moment comment on where this fits into the, there's some discussion that, RNA-seq is replacing microarray technology in some cases, and certainly not from a clinical point of view yet, but from a research point of view and where this sort of fits in and how this might adapt to that. I know that may be a very large question, but maybe just a sentence or 2 on that.
I don't know that I can contribute much on that, because as I look back on how fast technology changed in the last 2 or 3 years, if you'd asked me that question 2 or 3 years ago, I'd say, sure, it would be very important. But the fact of the matter is that I would have said resequencing on the microarray, like standard DNA resequencing on the microarray 2 or 3 years ago would have been a very feasible application of it, but it's not now. So other than the fact that I'm not completely knowledgeable about the technology that goes into the RNA-seq, I just feel that the technology is changing so fast that that maybe the microarray will be redundant, within a couple of years on it. It's going to be like Southern and like cytogenetics. It's going to have a very solid basis of use in specific areas, but technology in other areas is going to take some of the demand from it and put it into other technological streams. So, sorry, I can't answer it, basically. I was a long-winded, can't answer it.
Sarah, do you have one last question? Well, thank you very much, Christie and Karen. That was very enlightening.
Thank you. I apologize for going over time.